Hello everyone, I hope you guys are doing well and I'm back with another video. But this time the video is going to be about a topic that is related to drug safety or pharmacovigilance, and going over getting an overview about this topic will definitely help you in some ways, maybe in cracking a job interview for it, or maybe upgrading your skills, upgrading your knowledge related to the field of drug safety. So, without wasting any time, let's head to the video. Alright, as you can see on my screen, the topic for today is signal. Signal or signal detection in pharmacovigilance. So, what exactly signal detection is in the field of pharmacovigilance? So, we will be going through these definitions that are listed here later, but in general terms, signal detection in pharmacovigilance is a process in which potential safety concerns are identified in relation to any pharmaceutical product. And this is very crucial because for the protection of patient safety and ensuring the continued efficacy of drugs in the market, the signal detection is really important and plays a very important role. So, by detecting these signals, now what thing we get is we can take a timely action to mitigate the risks that are associated with that particular pharmaceutical product. So, if we talk about WHO, that is the World Health Organization, so according to World Health Organization, what signal is? Reported information on a possible causal relationship between an adverse event and a drug, the relationship being unknown or incompletely documented previously. So, according to the World Health Organization, it is any kind of information where there is a causal relationship. Now, by causal relationship, here we mean that there is a cause between an adverse event and a drug. For example, the drug is causing the adverse event. So, relationship here is unknown or incompletely documented previously, but there is a high chance, and there is technically there is a chance that there is a relationship between the adverse event and the drug. Next is EMA. So, EMA is European Medicines Agency, as you all know. According to EMA, a safety signal is information on new or known adverse event that is potentially caused by a medicine and that warrants further investigation. So, according to EMA, it is a safety signal, a signal that raises the potential safety concerns related to any new or known adverse event that has been caused by a medicine, and therefore further investigation is required. Next one is UMC. UMC, or the Uppsala Monitoring Center, Sweden. It defines signal is essentially a hypothesis of a risk with medicine with data and arguments that support it, derived from one or more sources. So, as per UMC, it is a hypothesis. Hypothesis that is something that is not proven yet, and a hypothesis of risk which is associated with the medicine based on the data. All right. So, what signal is? We have some information from different sources. Now, these different sources can be literature, it can be the spontaneous sources, and we get these information from the different sources, and there is a potential harm in the information which needs further evaluation, and there is a hypothesis that needs to be proven, the hypothesis between the causal relationship of adverse event and the drug or the medicinal product that is involved. So, when does signal detection start? Now, drug development has two phases. As you can see on my screen, there is a preclinical development phase and there is a clinical phase. Now, if we talk about the preclinical development phase, it is the phase in which a compound is identified and drug designing is done. So, here we have basic efficacy and toxicity studies. Whereas, if we talk about the clinical phase, there are few phases that are listed, which includes the phase 1, in which there are young healthy volunteers ranging from 20 to 100 and the pharmacokinetics is studied. Whereas in the second phase, there is the proof of concept, dose finding, and the selected patients with target disease. The range is 100 to 300. And the phase 3 is the efficacy, effectiveness, or safety studies. And in these cases, there are the selected patients with target disease, which range from 300 to 3000. So, based on what phase it is, we categorize it on the basis of what volunteers, what patients there are to be studied, what is going to be the number, and what is the basic parameter that is going to be studied in various phases. The next part is the exclusion criteria. So, as you can clearly see from the word exclusion, what exactly exclusion is? Something that is excluded. So, excluded or that does not qualify for that particular thing. For example, we have here polymorbid patients, children, elderly, pregnant women, women of childbearing age, long term treatments, renal and hepatic failure. So, these are the various people, or you can say the population, that is to be excluded during the signal detection because they are vulnerable and therefore they're excluded. As you already know that even during the clinical trials, these are the participants, these are the population which is generally excluded. So, why is signal detection needed? Now, post marketing, and if we talk about post marketing, the phase 4, it has extension studies, new indications, new formulations, new dosage, selected patients. So, the scenario is that even if a drug is in the market, it doesn't actually qualify for complete benefits. There is a high risk that the particular drug do have some risks associated with it always. So that is why signal detection is always needed. And in order to identify the emerging safety issues with medications, there are few which are not detected during the clinical trials, and for this reason, signal detection is therefore needed. So, this ensures there is a perfect benefit and risk balance of the drug, which is continuously monitored and the protection, the patients are protected via various actions. So, in order to just talk generally about why signal detection is needed, we can talk about various points, which includes the very first one as it helps us to identify the new adverse events. Secondly, it helps to monitor for the changes in any known reactions. Third, it helps to detect drug food or drug alcohol interactions which are not into the limelight during the clinical trials. The other part is it helps in recognizing the medication errors or the product issues. Then it also helps in identifying the new populations that are at risk. It also helps in detecting the new beneficial effects as well. So that is why signal detection is really an important part. Now, what can be the sources of signal detection? So there are various sources. We get the ICSRs from various sources, and therefore, if we talk about the signals, uh, the sources of signal, WHO program for international drug monitoring is there, and there are various sources that are listed, which is VGBase. So VGBase is a platform where we receive a high number of data that is related to the adverse events, and it is a WHO global database. So the second one is the medical literature. Third is the website of drug regulatory authorities. For every country, there are various drug regulatory authorities where we can get the signals from. Then we have the general media. In general media, we can include the various social media websites. For example, at times people reported through social media regarding any of the adverse effects or the unusual effects that they encounter after taking a particular medication of a particular company. So these can be the sources of signal. Now, what actually triggers a signal? So unexpected ADR is the very first point, which is the adverse drug reaction that is completely unexpected and it is not into the limelight, it is not detected during the clinical trials or the drug development phases. Other one is the new aspect of known ADR. Now, what is the new aspect? For example, there is a drug that causes anemia. We know that this particular drug is going to cause anemia in the patient, but there are rare chances, and we get a signal which says that the anemia is a pernicious anemia. Now, this is where a unknown aspect of ADR came into limelight because that is a particular category of anemia. The other thing is it is life threatening or fatal outcome. Now, if a signal is triggered, if a person takes a drug and knowing that it is not having any fatal effects on anyone, but a death is encountered after taking a medicine, then this triggers a signal, which is a major signal since it is leading to the death of the patient. Now, any other specific ADR, any clusters, any media. So, clusters here refer to a group of disease occurring together after taking a drug that generates or triggers a signal.